Okay, so today we are going to talk about the probability and the distribution. So this will solve the problem regarding to the statistic modeling. So state modeling or generative models. So this is especially relevant to this age. We have a lot of generative AI, so this all the probability and what's the next word before you have a sentence, what's the next token afterwards. So they all generative, so it means you have a underlying model, so you know what is the distribution supposed to look like based on your data observation, based on your training data, and then use statistical modeling to help you to generate the next token, next word, next gene expression level in the given gene in the given cell. So we will discuss all the theory or knowledge behind this. As I said, machine learning is based on lineage algebra and probability statistics, so two foundations. And then if you want to go deeper, say computational statistics, you may need to know optimization procedures and how to search the space of parameter combinations in a more efficient way. But that's a computational side. We can just treat the optimization as a tool, as a black box, just use those tools. But this statistics and all the relationship between variables, how we describe the mean and the diversity, the dispersion of the data points, so they all built into this class, okay? So very quick introduction, another introductory lecture. So I hope up to today you have some idea and what we cover. I don't know whether it's fit your expectation to this course or you expect a completely different things from the beginning. But yeah, just quickly, we have all the foundations we need, and then we start to learn how to manipulate those data. In the same time, we will discuss, for example, today we will discuss the normalization problem, how when you have a matrix of data, what that means to do the normalization and what's the options we have. So they all related to today's lecture. So we are going to go through generative model and the concept and introduce or just cover several commonly used distributions. We will cover normal distributions, Poisson, negative binomial, and the T distribution. Then we will have descriptive statistics, how to compute the mean and the variance. Then we will spend some time to see a covariance matrix, because this is important. We have a distance matrix per previous lecture, but now it's a covariance matrix. It's between the relationship between variables. Those are the parameters describe your data points, not the distance between data points. This is a relationship between the parameters used to describe your data points. At the end, we will talk about this KL divergence. It's very important to compute the distance between distributions. Okay? By the way, your homework due by today, and some of you already submit, and most of you have done a very good job. You see how easy the homework is, okay? Easy to get 10 points. So we will be have a similar homework, another additional three before the end of the semester. So, yeah, will be slightly harder because, for example, you explain PCA, you may spend a longer time compared to explain matrix multiplication. Okay. Now let's start with the UMI count matrix. That's the real data. The data is my lab sequence. It's from a lymphoblastoid cell line. It's derived from a B cell. This is, of course, just a very small corner of this huge matrix. I just show a couple examples of the genes by randomly. So not sorted by the name of gene, not sorted by the expression level. And the cells are relatively homogeneous. They're all similar to each other. If you do a distance matrix, all the pairwise distance will be very similar, so you create some kind of cloud or ball of those. They're from cell culture. So what I want to pay attention to this is whether if you have never seen single-cell RNA-seq data, you may pay some attention to first, they are integers. Okay, they all count the numbers. Also, you know there are unique molecular identifiers. So UMI means you don't have a same copy, and you count same copy. You count twice. They're just, if you have 3 of this gene in this cell, that means you really have 3 different copies of that. Of course, 3 is just approximate of the concentration or abundance of this gene in that cell. Then your next gene is much higher, it's 558. So that means this particular gene has been sequenced more about the different distinct copies. Yeah, let's go ahead. We're not good, so I'm using my laptop, but this is not given any signal if I switch back to the desktop. You see, there's no signal.
It says, so somebody changed the input on the monitor. Somebody switched the input on here.
Hmm.
So let's go back up. Somebody was messing with the inputs. Display.
Okay. Yeah, so I found this machine was booted. Okay, so in order to communicate with people about the distribution you build, you need some numbers or parameters. This is called the statistics, descriptive statistics, in order to describe the distribution. So we need first the central tendency. So you use mean, median, or mode in order to describe it. Or most of the frequent observation you should expect from the whole distribution. So another is you need, most important is variation or dispersion. So this you can use variation, standard deviation, or range of your data. So this, the most important descriptive statistics. So what do spread of your score or your number look like? So this is where the dispersion is important. The frequency and the positions as well. So those will be used to do the hypothesis test. So if you have a t-test, this test, they all rely on this descriptive statistics in order to compute some p-values. And you have an estimation and to get the parameters and the confidence intervals. Build this kind of a PDF, stands for probability density function, to display the distribution. So this is the range or bins of your data, and that's a frequency in your y-axis. So this is show you the different distributions have a different average, okay, different peak, and the different standard deviation, the variance. The larger, you have a broader shape, the curve, when you have a large variance. You also can have PDF. You can change that by accumulating all these values, and in your y-axis it become 0 to 1. This becomes a cumulated distribution function. It will tell you at a certain percentage how much your data will, at this cutoff, for example, 10, then you 80% of the beta point has been covered when you have this given value in the x-axis. So that's from a PDF, you get a CDF, okay? From PDF, there's PDF and this is CDF. We to visualize the distribution, we can use box plot. Box plot, you have a median, which in the middle, then you have a 25 percentile, 75 percentile. This is interquantile range. So this is a very important concept. So you can use interquantile range to describe the dispersion of your data. Well, the greater than 25 percentile, the data points will be located in here, and then 75 percentile lower will be here. Those half of the data points should be inside this box. And above that, you should have a minimum value, and before you have 1.5 away from the interquantile range. So this bar indicate the values which to help you to get this particular range, and anything out of that can be considered as a potential outliers. Okay, you point this out. So this gives you a very comprehensive description of the distribution, including the central tendency and also dispersion of the data points body. Then you can put them together. You have a CDF, PDF, CDF box plot, which can help you fully understand the distribution, the shape, the bias, the outlier distribution, and the range, So this, for example, is a very standard, and you have this shape of this box plot. And then if you have a wider range of this plateau, and then you have this very flat increase, okay, not a steep. And this is CDF, you can see the shape, and you can have a wider box in the box plot. Well, if you have a peak in the PDF, then you have a sharp jump in the CDF, and you have a very narrow box because majority of data is within this narrow range, so you have a box plot very narrow. And if you have this long tail and the peak shift to the left, then you have CDF look like this, and your CDF also have a long plateau in the region which close to 100%, the frequency 1, and your box plot, it will be shift to one end. So just look at them distribution, and if you have a comprehensive understanding, you may want to use all different kinds of technology to visualize your data. And this gave you twin peak, and you can barely see the difference, and this information is completely lost. If you use a box plot, you don't see these two peaks. So sometimes you want to visualize your data, use different technology in order to capture the different perspective of the feature. Okay, so it's very easy to compute the significance. If you have an accurate estimation of your data, you have a good model to reflect the true distribution of your data, then you can compute. For example, if you observe something in this region, you know how likely you should see it, and based on this PDF, and you would consider the region observation. If you located in here, and you have a certain cutoff that the probability of the observation is lower enough, then you will claim it's significant. Okay, so if you have this, you can have the. If you have a very complex, some distribution look like this, and it will be hard to see if you have observation here. So which p-value is supposed to be. We always may encounter some kind of standardized random variables. This is called a z-score, and to compute the z-score, you will have to use every single data point. So x, so here is a list of x1, x2, x3, xn. So this particular x is a variable, and the x contains n numbers of values. So now we minus its average. So this is a single value because average, this can be computed. And so I will use xi minus average, and then we normalize divided by its standard deviation. So you can use this array of data to compute sigma standard deviation. So to normalize this whole vector, you subtract every element with its mean, and then divide by the standard deviation. So this z-score is for every element in your variable to become kind of standardized, okay? So after you compute this, the distribution of this will be centralized around the 0, and then the shape. You have a mean average equals 0. Okay, so this is a centralized distribution of the thing. So this is a very commonly used standardization approach for your data. So I will quickly mention the multiple testing correction. So our why we need that. Okay, so this is a problem. So in the genomic data analysis field, when you have enough samples, before you only have one or two the classical experiment design, but now in the genomic and the transcriptome time, so we have many genes. If you do some test, you may end up with repeat the same type test again and again. So in that case, we are reconstruct a distribution. So in that case, if you use classical cutoff, see, we have cutoff for significant 0.05, okay, 5% or 5%. What that means is if you have a normal distribution, then if you observed a value here, and it's a PDF, so we're talking about one end. So this probability is 0.05. If you're above this, the probability is lower than that, so you claim this is significant. However, in this case, is you only do this experiment once. So you for example, if you draw a bin here, so you go to the museum. Normally you see this bin machine in the museum. So you draw a bin here, and this bin jump to here. Is that a significant event? According to this one, it is, because the frequency you get here is really low. So your drop is a very lucky drop, is a highly significant event. Well, if you do this in the whole morning, you drop, it was empty. So you drop one bin, another bin, another bin. So you repeat this hundreds of times. And in one of this drop, you have that bin jump to here, okay? Is that significant in terms of the whole event? It's not, because you repeated the test many, many times. In theory, it will be rebuilding the whole distribution. So one of the bin you drop into here is one of the repeated multiple testing theory. So that particular test is not significant, because you, by chance, you expect to have several bins in less than 5% there, but because you repeat it so many times, you should have that in there. So that's why when you do in the genomic age, you have many genes or many tests, you need to recite your cutoff significant level. So if you do the experiment only one or two or few times, so this is one time, then you use original 0.05 as your cutoff. Well, if you do 100 times repeat, normally you will have to do divide by 100 in order to do a new cutoff. So if you have in 100 times, and one of the events reach to this much lower significant level, then you claim that particular test is significant, but not using the original, very relaxed cutoff. So if you have, say, 1,000, then you will use cutoff the 0.5 divided by 1,000. This is a new cutoff. So this is the idea behind the multiple testing correction. You select the new cutoff, depends on how many times you repeat your same experiment to reset this cutoff to be more stringent in order to claim one of the events you observed through the repeated testing is significant. It's called a Bufferoni correction. So this is a table. If you only do 5 times test your original setup 0.05, then you use 0.01 as your cutoff. Well, if you repeat 100,000 times your original significant 0.05, then now you have to use 5 times 10 to the minus 7. So people do this in the SNP test, in the GWAS analysis. Genome-wide association study, in this is a repeat test for millions of SNPs. Single nucleotide polymorphism, you repeat the test whether this SNP in the population is associated with a particular trait. And you repeat this, depends on 700,000, how many SNP you test. So in this case, you really need Bufferoni correction in order to reset a much stringent cutoff in order to claim the particular SNP association is strong enough to be considered as significant. So just very quick side way to introduce multiple correction. Okay, so we know all this distribution are related. If you change the parameter or reset the conditions, one normalization, for example, Poisson can change into normalization, normal distribution, etc., etc. So Poisson distribution is also very commonly used to describe these random events, the number of success in a certain range of the time interval. And this is also very commonly used. For instance, this is a examples. See, in a survey here, and you know on average you should expect how many cars will pass by here, and then you set up at that background and see, okay, in 1 minute, what's the car you expect to see pass this particular street or intersection. You have a long-term statistics, say, okay, for the whole year on average, you should have so many cars. With that background, which you set up a lambda, which is average. And then you will look at how many cars will pass in 1 minute. That's a certain time interval. And then you expect, depends on this average, you have a frequency to observe. For example, 15% you will observe 1 car pass. And this is a 2 pass. Then to compute this, how high this bar is, basically the frequency or probability to observe 1 or 2 or no car pass, given the background of lambda equals 3, you compute the Poisson probability using lambda as background. So on average, your long-term statistic tell you the average is 3. Then you have, if you observe 1, and what's the frequency, the likelihood you should have that. If you observe 2, then what's the frequency of that? So this is how to read this Poisson distribution histogram. And Poisson is a discrete distribution, so you always have a count numbers. So the shape of the Poisson distribution will change. The one we see like here is peaked at 3 and 2, and then go lower, you only see 12 car pass by a very low frequency. This shape is because you're given the parameter lambda equals 3. But if you have a lambda equals 10, then you have this particular shape, and you have a peak near 10 and 9. Well, if you have a lambda equals 4, you have this shape. And if you lambda equals 1, you have we will observe more than 35% of 0 or 35% of 1s, and then you have a quick drop. So you have this yellow curve. So the shape of this Poisson distribution is determined by a single number, single count. But you can use this to see, I believe my gene expression follow the Poisson distribution with the parameter lambda equals to 10. Then you can get this distribution looks like this, and you can, based on this, to generate that particular gene's expression in the data. So how can we do this? Statisticians use a lot of lambda in different scenarios. In the number of droplets, and then they have a lambda to simulate that. If you consider how many bins in the droplet, there's particular distribution. So many steps in the single cell IC, you can simulate this by using different parameters of the Poisson distribution. We are not going to go into the detail to simulate the droplet and those. We are going to use Poisson distribution, just directly simulate the number of gene expression level in any given cell. So we will have a function, and then we will have this function to generate those single cell data expression. Statisticians always observed more than expected zeros in the single cell data, so they decide to use so-called zero inflated model by adding extra bin in the distribution. For example, if I believe my data is follow the normal Poisson distribution, so if I believe my gene expression follow the Poisson, then if you have 0 here and 1 here and 2, this is a distribution that's high 2 bars, and you have. So I just follow this yellow one. So you have then you drop. So this is 1, this is 1.5, drop. So this is the kind of distribution I expect if I have, if I simulate a gene expression using Poisson distribution. With the parameter lambda equals 1. So I expect to have more than 35% of chance I get 0, and 35% get 1, and then I get something like this. So if I will pop up the expression. I will start to see, okay, this gene I want to simulate. The cell 1, cell 2, cell n. I will write 1 here, 0 here, 1 here, 1 here, 0 here. Sometimes I write 2, okay? 0, 0, 1, 2, 3, 0, 0, 1. So I pop up those numbers according to my model. Well, the statisticians say, okay, we can simulate this. Say gene 1, gene 2, very good, but most of gene 3, 4, 5, there are more than expected zeros in there. Those 0 numbers is more than I expected. So this bar is supposed to be higher. So Poisson distribution, lambda 1, not be able to describe this extra numbers of zeros. So they decide to add this portion. They just add this manually, basically, to construct a zero inflated model. So put these particular numbers, zeros in there to reconstruct a new model in order to let this distribution to generate something like that, okay? So you have a number of pi, a percentage. So on your left, on your right, so they're zero inflated, so you can see this bar, and they introduce a lot of zeros in this data distribution. Okay, the last distribution we will introduce is called the T distribution. We don't normally use the T distribution. The only attention I want you to pay is the difference, the shape between normal distribution and the T distribution. If I don't tell you the difference, you may look at, oh, they all bear shape, and if you draw it, you don't see the difference. Because I want you to compare the difference between these two. So what you have to pay attention to is this particular region. Okay. For the same combinations of parameters, the mean are all in 0 here. The difference between this is a T distribution have a, see if you believe this is foot, this is a seeker tail. So if you look at the normal distribution, it starts to drop, and suddenly you have very thin tail. But this T distribution gave you seeker tail. So that seeker tail means what? It means you have a higher chance to find the data which away from center here. So you have a more, compared to normal distribution, you will have more in that tail region. So if you generate a number, this number means the distance between cells. Okay, I can put a lot of them, have no difference, all class together. But if I generate a distance using T distribution, I will have more distance in 3, for example. Well, in the normal distribution, it will simulate there's no cells to have a distance between them equals or greater than 3. But you use t distribution, you will have a lot relatively in this region. So the generative effects, if you generate a distance from a normal distribution compared to generate the distance from a t distribution, you will find the data in t distribution generator will be more dispersed, so separated, because you have a lot of distance which greater than 3 or around 3 compared to normal distribution. That create effects of the more dispersed separation of the cell, which close to each other, even though they were close to each other. The effects of a t distribution is to disperse them a little bit. So you will see the applications in the t distribution in T-SNE plot. T-SNE plot is the cluster of the cell. If they're close to each other, in terms of distance, they will be close to each other. But if you simulate that distance using t distribution, you will start to disperse the cell, which very close to each other a little bit. So you start to see the detailed distribution in the highly similar cells. Okay, even though they have very small difference, if you use the t distribution to simulate their distance, you will have a more dispersed, slightly dispersed cell population than other models. So that's what the t distribution will create, some kind of effects of slightly increased dispersion between cells when cells are very similar to each other, are highly similar to each other, even though. Okay? So some mathematical description. So if you have continuous description of what the mean is, you use this formula, frequency and the differentiation, and then variation. Those are the tables for the commonly used distribution categories, and then the parameters. For example, Poisson distribution only use lambda, so the single number to describe the whole thing. Well, the normal distribution, you need to have mean and the variance, and the negative binomial, you need to have a R and a P. P is success rate, R is how many trials you want to get, how many success you want to get. Okay, so we have a mean, median, mode to describe the central tendency. Okay, if you have a different shape, then these three numbers will be different. So sometimes mean is more properly used in order to describe the central tendency. If you don't have a symmetric distribution, that's normal. And the mean will be used for the normal distribution description, the better. And we need to have some description of the dispersion, so how separate data points are. So the range, so from a minimum to the largest deviation, which the difference from the data point to the mean, okay? So the deviation. Variation, okay, we will use variation a lot, the sum of a difference from the data points to the mean. So you need to consider many distance. So if you have a distribution like this, say let's 2D examples. we need to take average from 2, then maybe center is here. Then in order to compute the variance, let's compute from this direction and that direction separately. You need to find the distance from this to the mean. So this mean is here, so you find the distance. So we compute x-axis direction variance. So you found all the distance to the center. These are the deviation. Then you sum them together. So you have a D1, D2, D3, D4 based on your data. So D, I put together, N. Then you normalize by how many? So the distance is supposed to be always positive. Yeah, so variation you need to compute in this way. And the standard deviation is a square root for that. Another important statistic to describe the dispersion is so-called the coefficient of variance. So remember, this is important. So this is CV. CV is standard deviation divided by mean. So if you compute, so this is variation. So sometimes -1 to standard deviation will be square root of this variation. So mean divided by standard deviation. So this is CV. No, the other way around. So CV is dispersion, so then you need to have a standard deviation divided by mean. So coefficient of variance is some kind of a normalized standard deviation, normalized by the mean. So on your right side, this is if you have something which data is generated from a Poisson distribution, then the CV will have a relationship with the mean in a linear form. So that means CV defined by standard deviation divided by mean, which is the square root of variance divided by mean. In Poisson distribution, variance and the mean is the same, so you can replace the mean with a square root of the mean itself. And this can be rewritten into this. So there's a relationship between mean and the CV if your data is from a Poisson distribution. Then you take a log from both sides, then this creates a straight line. So remember this, we are going to use this relationship to find the so-called the highly variable genes, because we expect for any given gene, if you have certain level of expression, you should have that amount of dispersion. So this is a fixed relationship. If you jump away from this straight line, you are special gene. So we will come back to this page when we talk about the highly variable gene detection. So this is a compute of variance, which is the equation I just write in here. So I put the distance between the data points from data points to the mean. So this can be rewrite as a deviation, which in that case we write xi minus the average. And then squared, okay, in order to get the variation. So this is the example. If you have a data point, say x1, x2, so you have a 17, 15, this is only 6 samples. The average of this is x bar 14. So every element minus this 14, they will have a positive and negative values. So this is a deviation. You squared them and sum. So this is a variation. Okay. So this number is normalized by divided by 5. That's how the variation is computed for x. X is a variable. This is called covariance. If you have two variables, so you know how to compute the variation. If I have only a variable called x, I can independently compute variable for y. Okay, there are 2, but I want to know their covariance. Okay, the covariance is defined by you first normalize your x, so you minus your average of yourself of x, then you normalize y to the self, then you have product between them, then you put them together. So this covariance describes these two dependence of those two. And these two are also, the covariance have a sign, so it's not always absolute value. So this is then normalized by the number of samples. That's the relationship between two variables. So that's the equation you can write. Okay. So this is the same as this, just some different ways, and using matrix operations in order to compute covariance matrix. Okay. In this case, we have three variables. In the example in the previous slide, we only have 2 variables. You compute xy covariance between these two, so there's only one number covariance. But if you have 3 variables, you compute xy covariance, xz covariance, yz covariance. So you need to have compute a matrix for covariance. So this is what the expression is about. So you have 3 variables, x, y, z, and their covariance is between them, so x, y, x, z. Right. So you get something at the end. So if you consider how this will be done, you will realize it's kind of matrix multiplication. Okay, if you have this somehow normalized into this normalized version, big A becomes small a. Well, there's only some linear operations in order to get average, and then every element you get normalized values. Then once you have that, the covariance matrix become a multiplication of two matrices. So it's a single operation will help you to get this. So you just need to spend some time to convince yourself. This is the same as what you can get from here if you do 2 at each time. So don't worry about this. We will be able to fully understand on the computers in computer lab, okay, to have this done. Scatter plot matrix in order to look at the relationship between variables. Okay, so those data points were described by 1, 2, 3, 4, 5, 6 variables, and you want to know whether any two variables are somehow related, and this looks like this variable and that variable is negatively correlated. So this is a way, one of the ways to look at the relationship between variables. This is a relationship between covariance matrix and correlation. So correlation tells you two variables, they are positive or negative correlated. And the correlation basically this standardized version of the covariance. So this can be computed interchangeably, and they're related. So this will tell you how to, from a covariance matrix, to get the correlation matrix. That's the relationship which is here. So divided by the standard deviation, XY's product. QQ plot, we will skip this. So the last is KL divergence. There's two people, the Kobark and Labrador. So this is very important. You will see this all the time. Nobody, maybe 85% of the machine learning literatures, they always say, I want to compare two distributions, how different they are from each other. So you have one of this red curve and there's a blue curve. There are two distributions and how different they are. So this is a KL divergence will measure the distance. It's based on the, you slice each bins and see how much overlap between them is some kind of entropy in there. And most important, the KL divergence, KL between distribution A and B is different from that KL divergence computed from a B compared to A. So it's not a metric, they're not not addable and not this flipped, not always the case. So remember, this is how the distribution, two very simple distribution P and Q, and only have 3 numbers each to compute the KL divergence. Okay, you will compute the frequency of this pairwise P and Q and take a log and then sum them together. If you compute the KL divergence with Q versus P, you get a different number. But that's the way how you compute distribution difference. Yeah, so this is called a Simpson's paradox. So if you believe this data points tell you variable X and Y are positively correlated, but if you look at them carefully, and the sub population will support the other way around. This, if you separate the total cluster, the data point into subclusters, you see the opposite correlation relationship between these two variables. This is the difference between probability and likelihood. So somehow, yeah. I think that's all for this week. Any quick questions regarding lecture and homework? No? Okay. See you Thursday. It's kind of windy outside there.